We're kicking off today the first talk of a series of 4 talks, and for these we're going to be sharing with you some very special scientists who've been working in our labs. They are middle school teachers who have been here working with us for the last 2 weeks, and this is their final week. And so they're part of our National Science Foundation funded Students Discover project. So let's hear it. Where are my teachers? Where are my teachers? And so today through Thursday, each day, one of the project teams is going to be sharing with all of us what they've been doing in doing science in our labs and how they're using that. They're going to then use this for lessons and connect to their classrooms to have their students engaged in real science and real discoveries. So today we have our first group. This is Team Dirt, who's been working in the genomics lab. And we have 3 middle school teachers. We have Jennifer Stalls, Taylor Fly, and Courtney Millis, led by Dr. Julia Stevens. And so I'm going to hand it over to them so they can tell us what they've been up to. Welcome.
Good morning. My name is Jennifer Stalls, and I'm was a 6th grade science teacher in Pitt County, where East Carolina is located. This year I'll be teaching 8th grade science, and these are my partners on Team Dirt, Courtney Taylor, and our leader, Dr. Julia Stevens. We have, for the past 2 weeks, going on 3 weeks, been working in the genomics and microbiology lab on the 3rd floor, looking at different bacteria and microbes that live in dirt. All of this is part of citizen science, or the Students Discover project through the Kenan Fellows program. So the 3 of us are 2015, 2016 Kenan Fellows, and our job is to take citizen science, which is the general public taking interest in a project. It could be anything from microbes, what we're working on, or birds, or shark teeth, or plants, and the general public is intrinsically motivated to collect this data and report it back to a professional scientist who is on that project. So our job is to see if we can take that model and then transfer it to our classrooms, where we have time constraints, we have children who may or may not be interested in the project that we're trying to implement, and how do we get our communities to participate in projects that they may not even necessarily know about. So that's been our charge for the past 2 weeks, and what we've been working on, trying to figure out how do we transfer all the fancy things that Julia does in her lab to our communities with regular, average, everyday products. Our project, we're looking at microbes, and specifically soil microbes. So to preface what Julia is going to tell you about her previous research from last year on the dandelion project, first I wanted to talk about, well, what is a microbe and where are they found. So microbes are these tiny microscopic organisms like bacteria and fungi that literally are everywhere. They're on us, they're in the air, they're in the dirt. Sometimes we can see them, sometimes we can't. And our job with this project is to study the microbes that plants recruit to their root systems. And Julia is going to tell you about the project that she started last year, and we are picking up on that this year with other plants.
All right, so I am a scientist in the genomics and microbiology lab, and we're just going to start with maybe a little bit of the science background behind this project, and then they're going to talk about the awesome and creative ways that they can implement this project in their classrooms. And so I'm focused on the rhizosphere of plants. And so what the rhizosphere is this area below ground around the roots. And so there's always a very important symbiotic relationship, so a beneficial relationship between the plant roots and the microorganisms that live there, so the bacteria and the fungi. And they do a lot of things for the plant. The microbes can get nutrients out of the soil, They can help them acquire water, so to suck water out of somewhat dry soil. And microbes can actually produce growth hormones, so like food for the plants to grow even stronger. And so that's the area that we're looking. So we're comparing the microbes that live on the roots, outside the roots, so that's called the rhizosphere, to the microbes that live inside the plant. So there are some bacteria and fungi that can actually get taken up by the plant and they live between the cells. And so that's an even closer relationship than the rhizosphere. And so we want to know, are these areas, are these microbes different than the microbes in the bulk soil, so this area that doesn't have root in it, and do the plants recruit their microbes out of the soil? And so why we think this might be the case is a study that I conducted looking at dandelions with middle schoolers last year. So last year, middle schoolers across the state grew dandelions in 40 different types of soil. And so I got interested in finding out if that relationship is common across all of the successful plants, those plants that we would call weeds, the ones that grow in cracks of sidewalks and on the roof of our building and in lots of really crazy places. And so that brings me to where we've been the last 2 weeks, on the roof.
So as you heard Dr. Stevens talk about, she wants to find more plants that have the same behaviors as dandelions so that she can continue to study them further and just learn more about how these plants are doing this and why they're doing it. So what we've been doing is extending her study and trying to design something where our kids can go and look for other plants with this pattern. So we've been practicing this week by walking around and finding all sorts of plants that grow in weird places, like the roof or in the grates along the side of the street. So this is right out in front of the museum. And we've just been kind of exploring these plants and taking them back to our lab to look at them further. We kind of put them in a little solution and then we grow them, which you'll see here. So here's one of the really cool ones that we found that glows. This type of bacteria is found in dandelion roots, and we know that it helps plants by producing antibiotics and also by producing plant growth hormones, so it makes those plants grow really fast and really strong. And it also glows, which is really cool, and our kids are going to be really excited about it. So what we've been doing is just trying to extend her study of the plant microbes by looking for more plants and more weeds that grow in all sorts of places. We've also been thinking a lot about how different teachers can do this that don't have access to the lab and to all that fancy equipment. So we've been also doing lots of experiments in the lab up there using household items to make all these materials.
All right, so talking about some of those household items, we've been exposed to a lot of fancy, expensive, rare equipment, but what we're trying to do now is trying to figure out what things all teachers, any teachers anywhere around the country, around the state, around the world, can use in order to do the same experiment so that we can go back to that citizen science piece so that the kids can give data back to Julia later on once we develop some of those lessons. So we've been boiling soil to get the nutrients out of the soil that the bacteria and fungi can eat, and then you can see them filtering on the plates. We've also used some agar, which is something used in Asian cuisine. It's kind of like a gelatin, but it helps solidify the plates that we are making. We're also using coffee filters to filter out some of our soil extracts. A really cool one that we did last week, we used a salad spinner as our centrifuge. We pumped it, and it was able to bring any of the solids that were left in the tubes to the bottom. Another cool thing that we've been using is a microwave. So on that side, you see an autoclave that we've been using to sterilize our materials, but no one has an autoclave. But you can get a microwave and sterilize it by just letting it go for about 5 seconds, literally. Letting it bubble and then letting it go back down. It's really cool. Also, for us to see specific bacteria or fungi, we're using tenactin, which is for like athlete's foot, and neosporin for cuts and bruises. And it's allowing us to see specific things within our samples. So basically the whole idea that we're using a lot of these grocery store items is, again, for any teacher to be able to use the same protocols that we are coming up with so that they can then collect the data on the sheets that we're creating so that Julia can have a ton of data sets so that she can come up with the rest of her conclusions. And here's a lot of our cool art that we made with those bacteria that we were talking about earlier. And that's it. Your team, Barrett.